Hi everyone, this is Fahad Mirza and I welcome you to the channel. In this video, we are going to have a look at EVO2, which is a state-of-the-art DNA language model. We are going to install it locally and then we are also going to generate a sequence which is related to DNA after getting it installed. But before that, let's have a quick look at what exactly this new model, because I believe that one of the most anticipated area when it comes to generative AI advancement is this biomolecular field. So EVO2 is a state-of-the-art DNA language model. DNA stands for deoxyribonucleic acid, which is a molecule that contains the genetic instructions used in the development and functions of all living organisms. So this model, what it does is that it models the DNAs for long context and also deals with their design. EVO2 models DNA sequences at single nucleotide resolution, which means in simple words that it can consider individual nucleotides, the building blocks of the DNA, one at a time. And it can do that up to 1 million base pair context length. EVO2 also uses the striped HYNA2 architecture, which is a specific type of neural network architecture designed for efficient and scalable processing of large dataset. And this was pre-trained using this Savannah framework, which is another open source framework for alternative architectures. Now, this EVO2 was trained autoregressively on OpenGenome 2. OpenGenome 2 is a massive dataset containing 8.8 trillion tokens of genome data from a wide range of organisms that provides a comprehensive foundation for training and testing AI models. So that's a quick intro of this model. Let's try to get it installed, and before I do that, let me also give a huge thanks to Massed Compute who are sponsoring this VM, which is Ubuntu 22.04, and my GPU card, NVIDIA H100 with 80 GB of VRAM. Now this model comes in two flavors, 40 billion and 7 billion. Both of these models require you to at least have NVIDIA H100 or similar sort of GPU. Even the 7 billion one, I think there is some architectural limitation there which they are working on to improve. And 40 billion one requires multi GPU cluster. So I'm just going to go with 7 billion one, and we will get it installed. Also, if you're looking to rent a GPU on very affordable prices, you can find the link to the website of Massed Compute in video's description with a discount coupon of 50% for range of GPUs. So first up, let me quickly create a virtual environment with Conda. Make sure that if you're following along, you also install it at least 3.11 Python. And next, let me install the prerequisite, which are torch and transformers. Okay, next up, let's git clone the repo of EVO2, and I will also drop the link to it in video's description. Let's wait for it. It is slightly larger repo with a lot of sub folders in it, so it is downloading everything. And then we need to install all the requirements from the root of the repo. And while it does that, let me also introduce you to the sponsors of the video, who are AgentBot. AgentBot lets you effortlessly deploy a personalized knowledge bot across platforms like Discord, Slack, and others. It is ideal for open source tech communities and startups that provide user support, and I will drop the link to their website in video's description. And meanwhile, everything is installed. Let's download our model. In order to download the model, you would need a reach token for Hugging Face. So you would need to go to huggingface.co, and then from there you would need to grab your free read token. You don't have to pay for anything. And that token is available from your profile. So I'm just going to paste it here, press capital Y, and then it should log me in. As you can see here, let me clear the screen, and now let's download the model. I'm just going to use the Hugging Face CLI, and there you go. So I'm just using downloading this EVO2 7 billion model in a local directory which is called as checkpoint. And the model is not that huge, I would say, considering its GPU requirement. So you can see the model is just under 14 gig. So make sure that you have that much space on your system. So let's wait for it to get downloaded, and you can see that the download speed from Massed Compute is quite good today. And the model is downloaded. Let me now launch my Jupyter notebook, and then we will do the sequence DNA sequence generation in browser environment with this model. Okay, so the Jupyter notebook is here. Let me show you how you can do the inference with this model. So first let me explain this code. So what is happening here is that with this code, EVO2 is generating DNA sequences based on the prompts. So first up, we are importing the EVO2 module which we have just installed. Then we are specifying the model which we just downloaded, and from there we are when we say model.generate, we are generating DNA sequences. So there is a prompt sequence, as you can see, which is ACGT, which I will explain shortly. Then we have number of tokens, which is the length of the generated DNA sequence, which is 400 in this case. And then we are specifying temperature to 1, which controls the randomness of the generated sequence. And then we are specifying top k as 4. So this is the number of top candidates to consider when generating the next token. And then we are simply printing out the response here. Now let's talk about this ACGT first. Look, I'm not a any scientist or anything like that, but my little research shows me that this ACGT in the context of DNA sequences is refers to 4 nucleotide bases, and again nucleotide is simply the fundamental block or the component of DNA. So this ACGT are 4 nucleotides bases that make up the DNA. So A stands for adenine, C stands for cytosine, G stands for guanine, and T stands for thymine. These 4 letters are the fundamental building blocks of DNA and are used to represent the sequence of nucleotides in a DNA molecule. So here in this code, we have put the ACGT in a list, and it is being used as a prompt sequence parameter, which means that the model is being prompted to generate a DNA sequence starting with the nucleotide adenine, cytosine, guanine, and thymine. So let me quickly run this. And there you go, it has generated a long sequence of DNA nucleotides, ACGT, and this is based on the prompt which we gave it above in the code and the model's training data, of course. So look, I cannot explain it further what exactly this means. Maybe if you're a um, DNA scientist or someone like that, please also give us your opinion, especially as how useful it could be in generating this DNA sequence. Because for me, I believe that this could be a real game changer when it comes to maybe something like protein generation or something like that, maybe in some gene design or synthetic biology. Maybe I'm thinking vaccine development or some sort of drug discovery, or maybe crop improvement. And if we can generate sequences from about DNA from the models, that could be a real game changer. Anyway, let me know what do you think about it. If you like the content, please consider subscribing to the channel. If you're already subscribed, please share it among your network as it helps a lot. Thank you for watching.